To see things in the seed, that is genius. The ancient philosopher Lao Tzu recognized that the massive potential of a whole forest is contained within a single point. Look inside your kitchen pantry right now. Those dry objects sitting in your jars are biological engines currently on metabolic standby. They are waiting for a specific set of environmental triggers to initiate a high-energy deployment. This state of suspended animation is incredibly durable. Researchers recovered seeds buried in the dry lake beds of China that were carbon dated to be over 1,200 years old. When finally exposed to moisture, they sprouted as if they had been harvested only yesterday. To understand this survival, we have to map the seed's anatomy, track the metabolic acceleration that wakes it up, and look at the theory that plants have utilized these specific traits to influence human behavior for thousands of years. The seed is the ultimate biological vessel. It is the bridge between the final stage of a mother plant's life and the beginning of a new generation. The process begins when a mature plant finalizes its reproduction cycle, casting the seed off to survive as an independent organism. This chart shows the specific nutrient payload packed into the casing, precise ratios of vitamins, minerals, proteins, fats, and carbohydrates. But this internal inventory is chemically inaccessible. The seed maintains metabolic inhibition, preventing consumption until the environment crosses a specific moisture and thermal threshold. Breaking this inhibition requires 3 exact inputs: a specific volume of moisture, access to oxygen, and a continuous temperature range appropriate for the species. If the soil is too cold or the moisture level is insufficient, the chemical inhibitors remain active. The seed stays asleep. This prevents the organism from wasting its finite energy stores in an environment where it cannot survive to reach photosynthesis. Dormancy is a calculated waiting period. The seed is a biological sensor, scanning for the exact inputs required to guarantee a successful launch. When those requirements are met, water becomes the primary catalyst. It breaches the outer hull and dissolves the chemical inhibitors that hold the embryo in stasis. The presence of H2O immediately triggers the synthesis of enzymatic proteins inside the seed. This graph illustrates the scale of that reaction. These enzymes create a million-fold acceleration in metabolic chemical reactions, turning the seed into a high-speed refinery. In the first phase, enzymes convert complex carbohydrates stored inside into simple, singular sugar nodes. These sugars provide the immediate, easily accessible fuel source the seed needs to finance the rapid expansion of its cells. Simultaneously, a second enzymatic process begins disassembling stored proteins into individual amino acid building blocks. The embryo uses these amino acids as the raw material to build new structures, like roots and leaves. Germination is a violent transition, a massive spike in the speed of chemical deconstruction and biological building. To see how this chemical refinery is physically organized, we can look at the anatomy of a standard kidney bean. The outermost layer is a protective shell known as the testa. The testa's job is to shield the embryo from physical damage and ensure moisture doesn't reach the interior until the conditions are right. Inside that shell, the bean is split into 2 halves, called the cotyledons. These act as the first leaves, but they are primarily storage tanks for the carbohydrate and protein payloads that power the initial growth phase. As the plant grows, it draws mass directly from these tanks. You can actually see the cotyledons shrink as their fuel is transferred into the new plant. The seed's anatomy is designed to prioritize the protection of this fuel supply above all else. The true embryo sits between these 2 fuel tanks, ready to deploy in 2 different directions. The first structure to emerge is the radicle. It pushes downward, driven by gravity. This becomes the primary root system, tasked with anchoring the plant and sourcing external water once the seed's internal supply is exhausted. Almost immediately, the plant fires its second vector, the plumule or shoot, which travels upward. The plumule breaches the surface to reach the light. Once it does, it transitions the plant from consuming its own stored fuel to producing energy through photosynthesis. Biological sprouting is the successful execution of these 2 opposing vectors, one solving for water and stability, the other for light and air. Plants face a unique challenge. They are stationary. They cannot move to find better resources or spread their offspring. To survive, They have to outsource their movement to external agents like wind, insects, or animals. Author Michael Pollan suggests that crops like corn, apples, and potatoes have used this need to influence human history. By evolving nutrient-dense and flavorful fruits, these plants encouraged humans to spread their seeds across the globe. We have become the primary agents for their global dispersal. We have dedicated millions of acres and massive amounts of labor to ensure these specific genetics are protected and replanted every season. We believe we are the masters of agriculture, but the biology suggests the seed's evolutionary strategy has successfully persuaded us to work for its survival. In modern hydroponics, we take this relationship a step further by removing the unpredictability of nature. Natural soil is inconsistent. Its temperature and moisture levels change constantly, which can cause seeds to remain dormant or fail. To gain total control, gardeners use precision mediums like rock wool. These are sterile, inorganic materials that provide a perfectly uniform environment for the roots. By using these mediums, a gardener can ensure every seed receives the exact hydration and temperature it needs to break dormancy simultaneously. This bypasses the waste and uncertainty of traditional farming. The gardener provides the exact triggers the seed's biological engine is looking for. In this controlled environment, the radicle can push through the fibrous wool without resistance, and the plumule can rise directly toward artificial light. This isn't changing how the plant grows, it's providing a perfect set of inputs for the plant's existing biological programming. Once the seedling is established in the rock wool, it is moved into a high-tech fluid delivery system. This allows for continuous growth cycles that are not limited by the seasons or soil quality. This process turns the individual seed into a predictable unit of production. By mastering the unit, we can scale this system to create highly efficient vertical food networks. Understanding the anatomy and chemistry of this biological engine allows us to produce high-density yields with mathematical consistency, regardless of the outdoor environment. If you want to keep exploring the science that powers our natural world, subscribe to Unearth the History. Leave a like, hit the bell, and comment on what biological mystery we should unearth next.